Welcome back. We're going to start with our fifth and last workshop in a minute. It's going to be on Transcranial Direct Current Stimulation. And as usual, please ask all your questions on Slido. So please welcome both Lorenzo Diana and Professor Bolognini. And with this, I leave you the floor.
Thank you. I'm very, very happy to be here with you in person. I think Nadia really wanted to be here, but she's definitely following and she's having a talk in the afternoon. I'm also very happy to share with you what I learned with tDCS in the past years. So today I'm going to give you some practical hints. Today's workshop is divided in two parts. The third part would be a more theoretical one, with where I will try to answer some questions or to raise probably some other questions, and the second part would be more practical, hands-on experience with a volunteer, and we'll try a classical motor cortex montage. So on the right side, a little spoiler of tDCS, and this is just the outline. I'm going to introduce what tDCS is. We'll see together some of the mechanism of actions, practical stuff, current intensity, current duration, electrodes, and we'll also see something about the safety and some clinical evidence. And I will conclude with just a recent meta-analysis about tDCS and phantom limb pain that Nadia Bolognini in the afternoon will tell you much more about. So what's tDCS? As the name suggests, is a non-invasive brain stimulation technique that is, we do not implant any electrode in the head, but we just place them over the skull, and it was shown to modulate the excitability and to some extent also the plasticity. Okay, and this is done by means of a low constant electrical current. In a very easy setup, we use just two electrodes, anode and cathode. In the case of an anodal stimulation, we put the anode over the area of our target area and the cathode over a reference area, and the current enters the anode, exit the cathode. This is like the very, very simple explanation. tDCS is part of a bigger family of transcranial electrical stimulation techniques. As I said, it uses a direct current, as the name suggests again, but other possibilities are also available, like tACS, which uses a sinusoidal current, and it's thought to entrain with specific brain frequencies, but also the random noise, which is a kind of tACS. I just looked up on PubMed some days ago as I was making my presentation how many studies were there about tDCS, and I found over 4,000 studies. So tDCS has really stimulated, attracted the sorry for the joke, that was not meant. It really attracted the interest of many, many, many people, and it was used to really to shape and to induce modulation of very primary cortices like the human motor cortex, but also to modulate higher cortices and higher functions like social cognition, for example. From the clinical point of view, really a wide range of application, from treatment of depression, rehabilitation, from like stroke, both on the motor side, but also like aphasia, language disorder after stroke. Also to prevent cognitive decline, as you can see, and drumroll, also it has been used with phantom limb pain and sensation, to treat phantom limb pain and sensations. But how does it work? So we know that tDCS is able to modulate the membrane resting potential, and by means of hyperpolarization and depolarization mechanisms. And here there are some very interesting seminal studies on the motor cortex. So here they just applied 5 minutes of anodal or cathodal stimulation on the motor cortex. Then they used TMS, so magnetic pulses, to record the motor evoked potential, okay, as an index of the motor cortex excitation. And what they found is that with anodal stimulation, so the upper line, the motor cortex was more excited after 5 minutes of stimulation, as indicated by the increased motor evoked potential. On the other hand, with cathodal stimulation, the motor evoked potential amplitude decreased. And you can also appreciate that the effects, well, here maybe not very long lasting, but at least they last for some minutes, only after 5 minutes of stimulation. So they also wonder whether there were long-term depression, long-term potentiation mechanisms associated with this effect. So from that study on, there were a lot of neuropharmacological studies where they administer like an antagonist or agonist drugs that could be involved in long-term depression, long-term potentiation, and they saw whether they could modulate these after effects that we saw. And in fact, there are many like neurotransmitters and receptors involved. I will not go into details, so you will just trust me. For example, a sodium channel, calcium channels, like involved in the, oh my God, yes, exactly, in the hyperexcitability or to lower excitability of the cortex, but also long-term potentiation mediated receptors such as the MDA of the glutamate. This is a very interesting work that I really recommend reading if you're interested in knowing more about the mechanism, because it's not just like LTP, LTD. I mean, there is really much more also related to the gene activation, gene regulation, the expression of proteins, and also the glial cells. Okay, so let's see something practical now. Okay, what about the electrodes? We usually use rectangular or square electrodes. They are by far the most used. There are also circular ones. We just put them into saline solution soaked sponges, okay? And that's a very basic, super basic setup. Of course, the spatial resolution is not the highest, but it is what has been done mostly so far. But that's why some people wonder, can we increase the spatial accuracy of the stimulation? And the answer apparently is yes. So there are also other electrodes available, like silver chloride electrodes that can be used with gel. They may require some specific hardware, but there are a bunch of studies showing that, so like in the upper part of the figure, you see this 4x1 montage and how the current is really focal on a specific area of the brain, instead with a classical anodal cathodal, like here, classical conventional montage, the current really spreads all over the brain. These are just simulations. There are also other kind of electrodes available. This is one, it's a picture I took during one experiment of mine. These are concentric electrodes. So again, they're made of rubber. There is a small circular one in the center and a ring-shaped one, okay? They can be used with conductive paste, so the application is also very easy, and you do not require specific hardware in order to use these electrodes. This is just I just wanted to show you this very interesting study by Cancelli and colleagues some years ago, and here they even created personalized electrodes. You see here it is a bilateral anode. Okay, it was based on the individual brain, the individual MRI, okay, and apparently using this kind of setup, so these personalized electrodes, they were able to reduce the fatigue in a sample of multiple sclerosis patients. That's just an example. So where do I put the electrode? How do I localize the area that I want to stimulate? Well, one possibility is to use the 1020 system of the EEG. So basically, which refers to the classical positioning of EEG electrodes. You can just measure the distance from, say, the nose, the nasion, okay, to the inion, and basically moving 20% or 10% using that distance, you can put your electrode according to that measures. This is largely employed. It's very easy. I'm going to show you afterwards how easy it is. It's not the most accurate, but if you want to know what area you are actually stimulating, and you cannot run a simulation, you might want to read also this paper when they really basically they projected the location of the electrodes onto the brain, and they provide you a list of what area you are stimulating if you're placing your electrode, say, on C4. Okay, that's very interesting. There are other methods which are much more accurate, but also a bit more expensive, like the neuronavigated localization method. It's largely used with TMS, and this is especially for clinical studies. Someone argued that this is actually the best because it can take into account the inter-individual variability, so you really navigate the brain of the subject according to stereotaxic coordinates. What we usually do anyway, what I would recommend, is to perform a simulation of how the current spread. Better if you have, like, for every participant an MRI scan, but it's not always possible, of course, but you can use some templates. There are some free software like NIBS. This is just an example of a simulation we performed for our study. We wanted to stimulate the posterior parietal cortex, and as you can see, we were actually stimulating like most of the right hemisphere, but yeah. And just to show you that this localization story is not trivial, here these authors compared basically different methods to localize the dorsolateral prefrontal cortex, which is a very important target to treat depression. And they show that like different methods actually led to different localization of the area, but when they perform a simulation, they also found, of course, a different carry distribution. So depending on how you find the area that you want to stimulate, well, I mean, you could actually be stimulating different areas. So that's also quite important. So where do I put the reference electrode if I'm using two electrodes? Like usually one classical position is the contralateral, so on the other side, on the other hemisphere, just above the eye, supraorbital position. We may want to use like larger electrodes so that the current density is reduced, okay, so that we don't have an effect on that area, because anytime, let's say I'm performing anodal stimulation on the motor cortex, I'm also performing cathedral stimulation on the orbital prefrontal cortex. So I very much like this quote, that is, the fact that tDCS creates an electrical field using both a cathode and an anode is a blessing and occurs at the same time. There are also other possibilities that were shown to avoid this problem of multiple, let's say, stimulations. So you can put your reference electrode in some other parts of the of your body, okay, like the chin, the cheek. Okay, many have been tried. Maybe a bit more difficult to model the current distribution, but like these authors did, so it's possible. Something about the intensity. The usual current intensity ranges from 1 to 2 milliamperes. Usually you want the current to ramp up for some seconds, okay, so that the participant adapts a little bit to the sensations on the head, okay, and then it stays for the amount of time you desire, and then it ramps down again. So you may wonder, how do I choose the right intensity? And one could say, okay, you could replicate other studies. But then sometimes, of course, it could make like a vicious cycle that everybody does the same thing without even testing whether that's effective. So ideally, we should really test the dose response effects, and this is super relevant for clinical application, obviously. And if you're wondering whether higher intensity are associated with stronger or longer behavioral effects, one might say, well, I may expect so. The answer is that it's not always the case, because there is no linear relationship, and the intensity really interacts with the area I'm stimulating, like maybe motor cortex has a certain stratification, and other cortices are a different representation of the layers, but also the duration, okay, of the stimulation. This is just a very nice example, again, on the motor cortex. So the authors here compared 2 milliamperes versus 1 milliampere cathedral stimulation for 20 minutes. So we saw before that cathedral stimulation is expected to decrease the motor evoked potential, okay? And this is actually what they found with 1 milliampere stimulation, the lower line. But then when they applied 2 milliamperes cathedral stimulation, actually they found an increase in motor evoked potential, okay? So it actually resulted in an excitation. This is just to say that we should be careful. And also from the cognitive point of view, like these authors wanted to improve the working memory in schizophrenic patients, and they compared the effect of 1 milliampere and 2 milliampere current. And actually they found an improvement irrespective of the stimulation intensity, but then they ran some postdocs. And what they found is that the effects were stronger only after the 1 milliampere stimulation. So again, like the relationship is not linear. The same about duration. I mean, 5 minutes, 30 minutes, usually in the clinical application. For clinical application, it's about like multiple sessions of, let's say, around 20 to 30 minutes. But then again, would you expect longer effects with longer stimulation? Maybe, may not. I think by now you already know the answer, which is it depends. Not always. It's not always the case. Like, let's go back to the motor cortex, which is always very helpful. Like in this case, they stimulated with an adult stimulation for either 5, 7, 9 minutes, 11 minutes, and 13 minutes. Okay? So after 5 minutes, you see they were able to increase the motor evoked potential, but the effects were very short-lasting. So after 5 minutes, they already went down to the baseline. Then after 7 minutes, okay, the after effects were a bit longer, and then 11 minutes a bit longer again, and after 13 minutes, they were still able to observe a significant increase in the motor evoked potential that lasted for like until up to 90 minutes. Okay, so in this case, okay, longer stimulation, longer after effects, great. But then, okay, 13 minutes of stimulation, 90 minutes of after effects. And in a different study, they tried different combinations. So for example, 13 minutes and then another 13 minutes of anodal stimulation, and drum rolls again, so reduction of MEPs, so not even higher increase, which is very interesting. And then they try to apply a second stimulation during the time of the after effects. So 13 minutes stimulation, then I wait 3 minutes or 20 minutes, and I apply other 13 minutes of anodal stimulation, and they found that they were not really able to modulate, let's say, the short after effect, but the early after effect, but in fact, they could modulate the late effects. So they could see a modulation of the motor evoked potential until the next evening. Okay, so I think you can appreciate like the complexity of this technique and the underlying effects. Just a couple words about the placebo condition. Placebo is real, it works. I mean, but we should try to control for it. I mean, as much as possible, it leads to figure it out. And also, it has also an effect in the field of non-invasive brain stimulation. That's, I mean, it's so obvious, but I just wrote it that we should control for it. And that's why it's important to have what we call the sham tDCS. Usual protocols are like, okay, apply the current for some, let's say, 30 seconds, and then I switch off the stimulator, okay? So this is meant to just mimic the initial tingling sensation under the electrode, so that the subject believes that it's a real stimulation. That's not the only one. I mean, there are other possibilities, like like 2 peaks of stimulation, one at the end, one at the beginning, one at the end. Okay, there is a very large debate about what's really the best. Maybe what works with 1 milliampere may not work with 2 milliamperes again. So there are some nice review works published. Maybe there is one thing that I can say, like for sure, that tDCS is a safe technique when properly used. I mean, among these variability, I can say that tDCS is a safe technique. Like when we test healthy participants, we are very, very, very strict. We exclude participants with like migraines, head injuries, head seizures, or even familiarity with epilepsy and any sort of skin conditions or devices implanted. That's of course. But there is a review. There's a review published some years ago in 2016, and they gather a lot of data, and over more than 33,000 sessions of tDCS and over 1,000 subjects tested, they could not see serious effects of any kind or irreversible injuries. Okay, and so now the parameters that are widely accepted are that stimulations no longer than 40 minutes and not more intense than 4 milliamperes are definitely safe. Okay, and even if you repeat tDCS, so multiple sessions, which is again like classic for clinical application, you might want to stimulate your patient many days, more than 1 day a week, then again, it's safe. They didn't find a higher incidence of adverse effect. If you've never done receive tDCS, you may wonder what I feel. Okay. Well, nothing like just this itching sensation. You would like to scratch your head, but you can't, of course. And the intensity of these sensations are mild to moderate. Okay, so they're really very tolerable. And I always like to really collect and report in my paper with as much detail as possible all the side effects and the sensations. This is just a form that we use where we ask the subject to rate like itching, pain from null to very strong. There are some available in the literature. So just a summary so far. We've seen some of the mechanism of actions, LTP, LTD related neurotransmitters, hyperpolarization, depolarization, okay. We've seen the electrodes can be less or more focal and how to position them, okay. We've seen also the non-linearity of the non-linear effects of the intensity and the duration. About the intensity, like beyond, like around 4 milliamperes is okay. Okay, usual studies from 1 to 2 milliamperes. Duration no longer than 30 minutes is accepted. Okay. So now again, I think you're all wondering if it works. Okay, that's a very complex question. We've seen some results now on the motor cortex, so it does actually something in the brain. Good. But there are also negative findings published, like especially when we want to modulate cognitive functions and we want to modulate a healthy brain. But also from the clinical point of view, we find some effects of tDCS, but there are many reviews and meta-analyses reporting low quality, a lot of heterogeneity of these studies. Also, maybe not too many patients, and also the methods are not reported accurately. So again, a lot of heterogeneity. This study is published in 2017 by a panel of European experts about non-invasive brain stimulation, and they wanted to provide guidelines and recommendation whether I can use tDCS or not, okay, to treat some conditions. They consider really like a lot of clinical condition ranging from pain, Parkinson's disease, stroke, epilepsy, depression, schizophrenia, addiction, and they consider only sham control studies with repeated session of tDCS, okay, and they just provided the level of evidence according to the number of high-quality studies, like, okay, I found definite efficacy for the treatment, or just probable efficacy, but it's not a meta-analysis, okay. So they did not find definite efficacy for any treatment, so this has to be said. But the level B probable efficacy was true for fibromyalgia, or some aspects of depressive, the major depression, and for addiction with different montages, of course, mostly targeting, as you can see, like for major depression, dorsolateral prefrontal cortex. Also about the probable inefficacy for tinnitus and some other aspects of depression, and possible efficacy, just possible efficacy for chronic lower limb neuropathic pain. What they conclude is that we need a better characterization of all the parameters, but that also there was a large inter-individual variability. Okay, and this leads me here to this very important point. That is, we know that different subjects respond to tDCS in a different way. Some respond, some others do not respond at all. So I think the key question here is not really whether it works or not, but we have to figure out when it works and when it doesn't. So we really had to address this inter-individual variability. For example, okay, tDCS, we know that is a state-dependent technique. That is, if I apply tDCS during a task, okay, so if the task activates a given network and I stimulate an active network, I may see some effects. If I do tDCS offline, as we say, okay, so without any task, I might stimulate other networks and I may observe other behavioral effects. Like, for example, in this study, they compare online and offline tDCS. And they put also subjects into the fMRI, and they wanted to see the effects of tDCS on the brain connectivity. But so the focus mainly is online versus offline tDCS. And what they found, that's very, very interesting, that tDCS, when the subjects were at rest, they increased the activity of the default mode network, okay? And it did not increase the activity of the network they actually wanted to modulate, okay, which was related to the task. But that was the case when tDCS was applied during task execution, okay? So different effects. Also, tDCS is affected by many individual factors. One of them is like the brain structure and function. And this is again very relevant for a clinical application. This is a study I like very much, published on brain stimulation some years ago, and they wanted to use tDCS to improve depressive symptoms, okay, by targeting the dorsolateral prefrontal cortex. I'm a psychologist, by the way. That's why I'm interested in all like cognitive and neuropsychiatric disorders. And they found that some subjects actually responded, okay, but the larger improvements were seen in those subjects with larger left dorsal PFC volume, okay, gray matter volume. Also, another factor that is important that we should take into account is that the level of the baseline performance is also an effect. I'll just give you an example. So like in these studies, they wanted to use tDCS to boost short-term memory, okay? And they found that only in those participants with low short-term memory, tDCS actually helped, okay, to increase it. So this can apply to many different like situations. So that I should not expect that tDCS again has the same effect on every single participant the same way. So again, to sum up so far, we see the state dependency of the stimulation, that if I stimulate online versus offline, that is without or with a task, I may see different effects. But also we saw that the structural variability, but also the baseline level may affect my stimulation, okay. And finally, tDCS, as I said, was used to also to treat phantom limb pain and phantom limb sensations. I will not spend many words about that because then Nadia Bolognini in the afternoon will tell you a lot more about that. Just one possible mechanism that I found in many papers that I don't know tDCS, when applied to the motor cortex, so an excitatory stimulation could enhance the descending inhibitory pathways. So I will not say whether this is true or not. I will leave you like the decision, but this is a possible putative mechanism of action. And there is this very recent meta-analysis published last year where they compare invasive and non-invasive techniques to reduce phantom limb pain. And I will just focus on tDCS quickly. And they only included 3 randomized control trials because they were really strict for the inclusion criteria. Like no self-promotion here, but the studies were by Nadia Bolognini and colleagues, and but also Kickert. And just in general, the area that was stimulated was the motor cortex, or at least somatosensory cortices, because electrodes were put on the C3, C4 location, but they use, again, quite like conventional electrodes, okay? And they apply tDCS for one or more than one session, lasting 15 or 20 minutes, okay, using a pain visual analog scale. This is just like summary very in general, okay? And I hope you can read it. Like, they found an immediate pain reduction just right after tDCS, but also in 2 studies when they applied repeated sessions of tDCS, it seems that this pain was also reduced after 1 week. Okay, the average of 1.5 points reduction. But then, of course, this meta-analysis, as many other meta-analyses and review, concludes that we need more randomized controlled trials, more patients and longer follow-up, and that we should, like everybody should make an effort to report all the parameters like properly. There are also some open questions, which are still open, like whether more sessions do produce longer benefits, but again, we don't know because there were too few patients, and also whether if I apply tDCS online, So during an imagery task, for example, does it produce larger effects? So it may be, but it should be like demonstrated in further studies again. This is just to conclude that there were other studies targeting other areas that different from the motor cortex, like in Bolognini, they tried to inhibit the posterior parietal cortex, which is a hub for the body representation, body awareness, okay? And they were able to reduce the phantom limb sensations, but also the cerebellum. And Nadia will say something about this too. This is the very last slide. I would like to conclude with some must for the future studies. So on the one hand, I think we really need like a careful study design, randomized controlled trials. This might sound, I mean, this is obvious for clinical application, but in the field of non-invasive brain stimulation, I think we should really like make an effort in this direction. Also, the study should be well powered. It doesn't mean that I need 1,000 subjects. It means that I should calculate, like do a power analysis, like there are different like softwares available, but I should calculate according to the effect size that I expect. So how many subjects should I test in order to see that effect of that dimension? And improve, of course, the precision of the methods, compare tDCS alone and versus tDCS when it is combined with other treatments, okay? And also very important to study the dose response, because like I've seen really many studies saying, okay, like 2 milliamp per 20 minutes, okay, 2 must work, okay, but then we don't really know whether maybe 1 milliampere works better. And finally, we should really study and address the problem. Well, the fact that there are different responses for different subjects. So we should study the inter-subject variability, and we should explore also the predictors of the efficacy of tDCS. So if you combine, for example, tDCS and EEG, or with fMRI, you might see that, I don't know, like a given power in a specific brain frequency can predict the efficacy of tDCS, okay? So combine techniques. And yeah, that's all for the more theoretical part. Thank you for your attention. So if you have questions.
I think we have a question from our expert in...
Okay, I'm not expert, but you said tDCS, you can use it online with other tasks, but how do you think it helps the, like when it's used, when you use it with other, like the working mechanism aspect, I would say.
The mechanism, why it works better, apparently.
Yeah.
Well, so there's more than one study showing that, I think I showed also one before, that if you pre-activate the network that you want to stimulate with tDCS, like with a task, okay, then that stimulation is more likely to to work on that specific network, so to modulate more specifically that network.
So you would say it's going to be easier for firing?
Well, then so the biological mechanisms, that's a very good question, actually. I think I should look it up. I don't have like an answer right now, but that's a very good question, definitely. Because not really, like, sorry, we'll just add like a little piece. It's not like really the modulation of the area underneath the electrode. Like there are many other studies, well, maybe not that many, but some other studies where they use TMS, so magnetic pulses, EEG, to study the effects of tDCS, okay? And they show that actually the effects spread also all over the brain, like besides the models that I showed you before, but actually, so functionally in terms of network, the effect spreads. Okay, so that's probably also why in this sense.
Thank you.
Brooke Trainer from Richmond. So one of my backgrounds obviously is in intensive care, and I'm trying to find a good device to study. Eventually I want to study it because I think that long term it could have some benefits in our surgical post-op patients. But looking at the stimulation of the vagal nerve, So the vagus nerve is, obviously a vital function in a lot of pathways for, respiratory issues,
Yep.
gut issues, post-op ileus, and my thought is, would there be a way to stimulate the vagal nerve to be able to forego the need for all the pharmacologic options that we end up, routinely placing patients on in the acute post-op surgical period. So there's no non-invasive vagal nerve stimulator out there that I can find except for the one that the patient holds on themselves, to stimulate right over the carotid. But this, a lot of my patients are not able to do that post-op.
Yeah.
And I'd like to apply it, sooner than later and then study it. So would this be, just listening to your talk, it's making me think that, applying it over the supraorbital and shoulder, maybe I could target that same area, or has there been any studies looking at that?
So thank you for the questions. So I'm pretty sure that there are some studies where they try to apply it like, not transcranial, but non-invasive vagal nerve stimulation somehow. I cannot remember exactly whether they were effective or not. But yeah, when you apply, like for example, the reference electrode on a different position, there were some studies showing that you might have like some sort of other effects also including the vagal nerve, but other studies showed that, well, in the end, this is not a big deal. So like we would like to exclude this effect sometimes, but in your case, you would like to stimulate that. So I don't have an answer for now, but I'm sure that there is some research done about this.
Okay, thank you.
Okay, good. Okay, so where is the volunteer that kindly, please. Now I'll show you. Thank you. So I'm gonna show you a very easy montage for to stimulate the motor cortex now. I think it's, thank you. Okay, great. So just a couple things. So here I have my, oh, that's great. Like my 2 electrodes. In this case, I'm using 5 by 5, 35 square centimeters electrodes to replicate some previous studies. Okay, like sponges, which has already been soaked with saline solution. It's like in cooking show. I already done that. Okay, and now they're ready. Like measuring tape to take distances from the nasion to the inion, and then the 2 periauricular points. Okay, I'll show you. And then some bands, okay, rubber bands, in order to keep the electrodes in place. Good. And the very important, like whatever you want to mark, okay, the spot on the head. Great. So let's see how big your head is first. Okay, can you take your glasses off just for a second? Okay. So you see here, because we first want to find the center of the head, and then from that point we can move 20%, 10%, like anteriorly, posteriorly. Okay, that's why. Okay, so I'm looking for the occipital bone here. Okay, so it's like 37. Do you think it's, you have already participated in some tDCS experiments? Does it sound okay? So 37, the off would be 18 and off here. Okay. Great. Is everything all right? Yeah, okay, good. And then from here, just above the tragus, I can ask the participant to open and close the mouth. Can you open and close the mouth? Okay. Where I see, I can feel a little sort of depression hole here, and that's the right point. Okay. then again here. Okay. So here it's 36.5. Great. So here. Gosh, a little bit too much. Sorry. Okay. So this is CZ, so the center of the head. Okay. Do you think it's center more or less? Well, I think someone says that if you ask the participant, like they know. I mean, if this is the center, somehow. Okay, great. Then, well, actually, to find C4, in that case, I moved the 20% of the periauricular distance. Okay, so in this case, would be from this point here, so 20% in this direction, and I find the right spot. Oh my God, no, cannot make the calculation. I need technology. I'm sorry. So this would be 36.5. Okay, 7.3 centimeters. Great. So from the center to the preauricular point here. Okay. Okay. So around here. Yep. Just do it again, just to make sure. Yep. Okay. So this is the area, my target area. Okay. Then rubber strap here. Okay. Okay, usually secure it under the inion because the bone helps. Okay. Is it too tight? Is it okay? Great. Okay. Just kind of like here. I usually put the reference electrode first. Okay. For example, like here, so as I'm going to stimulate the right hemisphere, I'll put the reference electrodes in a supraorbital contralateral position here. I usually use like bigger electrodes, as I said before, so that the current density like is less. Okay. Great. Little gift for you. Okay. Then where's the other one? Okay. We're almost done. So here's the spot. So one pin here. Okay. Another one here. Great. All right. I try to remove the hair as much as possible. Tell me if it hurts. Sorry, I'm not very delicate here. Okay. Tuck. Okay. too tight? It's okay. I didn't pay her, I swear. I mean, like real subjects, real life. Okay. Okay, great. So this is one of many possible simulators, like conventionally, like red anode current goes out and comes back. Okay, so from the black one, but you should read the instructions. Okay. So it first performs an impedance check to see whether the impedance is too high. If it's too high, okay, so the stimulation won't start, of course. Okay, that's why we had the saline solution. Okay, let's see if it starts now. Okay, now it says, okay, I'm sorry, it's too high. Okay, so what we can do in this case is like to add some more solutions. Maybe we are syringe, like that you don't, it does, I mean, like the electrodes, sorry, the sponges should be wet but should not drip, because otherwise the currents goes again all over the head. Okay, let's see other currently used methods. Let's see, to remove some head first. Some hair, not the head, because if we remove the head. Okay, here. Okay, stimulation started. How does it feel? Is it like, well, painful? Hopefully not. What do you feel and where? Yes, exactly. This is exactly what most subjects report. This was very short stimulation. It is already fading out, but this was just to show you that, like in 10 minutes, you can be able to have your your montage, and it's quite easy. This, as I told you, you can use also more focal electrodes. I just brought them here to show you. Like also these ones. I mean, so compared to the square, well, that's very difficult to see probably, but it's around 2 centimeters diameter, and compared to the other one, it's much more focal. Okay, great. Here. Okay. And this is the return electrode. In this case, we use conductive paste. Okay, and that's also quite handy. Great. That's it. Thank you. Before, maybe. Okay, let's take it off. Okay. Oh, sorry. Better than the hairdresser, right? Okay.
I think your demonstration was so clear that none of the online viewers had any questions.
Okay.
So thank you very much for that.
Thank you again.
Thanks so much for that workshop, and then we will see each other again at 1:30 after our lunch break. Looking forward to see you both here in person and online.